The next speaker is, the next talk is actually from Kidney Precision Medicine Project. We had a slight change due to unavoidable conflict. Jonathan Himmelfarb was not able to present. So we are very fortunate that Becky Steck from University of Michigan will be presenting to give us an overview of Kidney Precision Medicine Project and metadata. Becky Steck is really the leader and the director of the software solutions in University of Michigan, and spearheading the data visualization center for the KPMP consortium. And and I regard her as really the glue and the control tower, who is really holding things from all the different directions together and making it available. And she will be presenting us the KPMP overview and the metadata approach. So thank you, Becky, and please take it over.
Thanks, Sanjay. First, just to test, can you hear me?
Yes, we can hear you very well.
And you can see my slides?
Yes, we can.
So apologies that Dr. Jonathan Himmelfarb wasn't able to join and do this talk, but I did find another one of the PIs from this project to come along. So this is Dr. Matthias Kretzler sitting next to me, and he's actually going to take the KPMP overview slides. So he's one of the 3 co-PIs of the central hub for the Kidney Precision Medicine Project, and he'll be able to explain a little more about what that means as we go on. So.
Yeah, good morning, everybody, and thanks a lot for giving us an opportunity to give you an overview of what we are planning to do in the Kidney Precision Medicine Project. Quick intro to myself. I'm a nephrologist and computational biologist, but I did my MD PhD in renal anatomy and cell biology with Wilhelm Kritz at the University of Heidelberg. So I had an early exposure to many of the opportunities and challenges who are surfacing now. And it was a fascinating morning for me. I was able to follow most of the morning discussions. This is really very exciting to see what the HubMap team is standing up. And just, as you will see in a moment, will nicely align and complement where we are with the KPMP project. The KPMP project, if you go to the next slide, has really a very specific focus. It's about understanding human kidney disease so that we identify novel intervention points to impact the diseases our study participants are suffering from. And the specific focus of KPMP is on the most frequent diseases seen in our patients with progressive kidney loss, which is acute kidney injury and chronic kidney disease, particularly diabetic kidney disease and hypertensive nephropathy. The goal, how we want to facilitate research, involves an assembly of a very comprehensive, deep understanding what the structural lesions inside the kidney in our patients with these diseases look like, and then to assemble this comprehensive multiscalar data set to generate a kidney tissue atlas. And this atlas will allow to identify critical cells, pathways, and targets for novel therapies in the individual patient-specific background. So linkage with the clinical phenotypic information prospectively ascertained from the patients is a critical feature of this effort. And then we expect to utilize that to redefine these diseases, which currently are syndromic, non-specific disease aggregations, into mechanistically distinct subgroups. And then to develop individualized treatments for the joint mechanisms detectable in the atlas among these patients, so that we improve the knowledge base and obviously feed forward the drug processing pipeline currently underway for chronic kidney disease. Next one. So what is an atlas? And I think we heard several definitions in this morning already. I think we define it here. That is a map who has multiscalar information already available as a kind of geographic, geopolitical, societal, and economic statistical features in present. Next slide. We envision the same to be needed to develop a kidney tissue atlas to be searched by our diverse user community. And KPMP currently has in scope to generate from renal biopsy tissue. So this will not be an analysis of entire kidneys. It will be from research kidney biopsy course-derived information using digital histological images, 2- and 3D tissue architectural mappings. We will develop cell states using a flurry of single cell and single nuclear technologies, and then wrap them up to link them into the mesoscale using the integrative approaches, and a wide variety of capture of the molecular state of the tissue using proteomic and metabolomic activities currently in scope and more to come. Next slide. The atlas construction will initially use a tissue to really capture the multiscalar knowledge we can extract from our patients out of the tissue, and we will have flanking biofluids available as well to see what non-invasively can be associated with the tissue lesion. Next slide. The key feature, however, then is that we will bring that information back together around the patient as the individual observation unit and correlate it with clinical outcomes and disease trajectories so that we can utilize that information to redefine in mechanistic terms the diseases of AKI and CKD, develop individual prognostic tools, and obviously pass for novel treatment development. For this enterprise, as you would expect, we have to have very unique and disparate expertise present and combined in KPMP. We have 6 recruitment sites focusing on acute kidney injury and chronic kidney disease to safely and effectively procure kidney biopsy tissue from patients who volunteer for research biopsies. This will not be as part of a diagnostic procedure, but really selected patients who volunteer the risk to procure biopsy tissue for research. We have 6 tissue interrogation sites who have developed a flurry of technologies using primarily tumor nephrectomy tissues over the last couple of years. Very carefully vetted protocols, including a comprehensive metadata set, which Becky will give you an update shortly. And then we have the integration units, the central hub, who's responsible both as a clinical coordinating center for the clinical operations of the project, and the data visualization core, which Ravi Iyengar and myself lead here, and where Becky is the key project officer to bring these informations together and make them visible. And we have an admin core for the overall logistics, including support of a significant opportunity pool, which is bringing expertise and resources and efforts currently not in KPMP to become integral members of KPMP. Next slide. We have established a biospecimen flow, as you can see here, where from the patient recruitment via central biorepositories, the tissue is distributed across the performance sites and then brought together at the central hub for the integration and display into the kidney tissue atlas. Next slide. And this was a quick summary. Are there any questions for the overall strategy and layout of KPMP, which I could address right now? If that is not the case, then Becky can dive into the data types and the metadata strategies we are currently deploying.
Thanks. Oh, good. Okay. So yeah, this slide is a nice one to park on for a second, just to show the different, the magnitude of the data that we're trying to collect from these biopsies. So we're going to be doing a variety of imaging information that we collected. As you saw in one of those earlier slides, a lot of different molecular data will, and as well as the way we've been calling clinical data. So just going to buzz through. Here's an example of the amount of different forms. These are case report forms that we are loading into REDCap for the longitudinal follow-up of our patients that are undergoing biopsy. You don't need to read all these words, but just to get the point across is there's a lot of them. We're collecting a lot of different clinical data. These next 2 slides are just to show the visit schedule. We're expecting to follow these patients for a very long time. 5 to 10 years, and they have slightly different schedules depending on if they were an AKI biopsy or a CKD biopsy. In addition, as Matthias pointed out, with all of the 3 different biopsy cores and then a variety of biospecimens, right, urine, blood, and stool, there's also a lot of tracking of all of that information. So there's a big lift when we think about metadata. There's a lot of data that we have just regarding what is the specimen and the provenance of that specimen. So they, the team at University of Washington has built a really great suite of tools between some of the data being collected in REDCap at the recruitment site during the visit, and then an additional in-house built tool called SpecTrack that's responsible for tracking the specimens. And each of these cores gets broken into different segments that get sent different places. So there is a lot of data that's being tracked there. But what I want to focus my time with you today on is more on the tissue interrogation site and their different, so in essence, our molecular and imaging data. So this is a quick slide just to show the different types of data and the different institutions that are involved right now in collecting this data. Hopefully, the point of this one is to say that it's a lot. There's a lot of people that are working on this and a lot of different great data that is being collected. But what that means is that we had to do a lot of work to try to standardize how that data was going to be collected and what metadata would accompany each of these data types, so that we could make sure that when we share all of this data, it's very reproducible, right? We've heard the FAIR standards a couple times already today. We've got that same remit, as well as just making sure that for folks that want to try and validate the data, that we have everything we need. So the way we went about determine that standardized metadata is kind of outlined here. So it started with, and apologies for the acronyms. So the DVC, that's the Data Visualization Center. So that is part of the group that is responsible for collecting and visualizing all of this information and ultimately building that kidney tissue atlas. So we started with the DVC team kind of deciding what we call here the pyramid of data elements. So, okay, what type of metadata needs to accompany? We need tissue processing data for certain types of our molecular experiments. We need analysis data. We need, right, so we kind of broke it into those major categories. Then we also, the TIS, that acronym is those tissue interrogation sites. So those are the performance sites doing the molecular analysis. They have, we worked with them to understand what metadata they were already collecting standardly, or what data they thought we should collect in KPMP. Sanjay is an example of one of the people that we worked with there. We also looked at some other bodies that have already, other consortium that have already done this. The LINKS consortium was one of the ones that we relied on quite heavily, since they'd already crossed many of these bridges for us. At that point, then we actually met with, we broke, if I go back a slide here, you can see, for instance, there's a variety of different institutions working on transcriptomics data. So we brought the key players from those institutions together, and we kind of went through our data pyramid and compared and contrasted and had some, the tough conversations to say, okay, we really need this piece of data, so you're going to have to go back to your sequencing core and get it, things like that. But then what is kind of neat about this is that after we did that step and we had agreed upon a set of metadata standards for the different technology types, we have a tool right now for all of our sites to upload their data to kind of a central repository that we call the data lake. So what we have built now is a system where the data lake can actually, sorry, this is some timelines here. This was all quite recent, and we just finished some of this last month. But the data lake can now read in this file where the metadata standards are kept, and present the user kind of a dynamic form based on which type of data. So it's kind of best to see it in a picture. So in this next slide here, I'm showing you, this is just a quick little extract of this file. So what you're looking at here is some of we have 3 different types of proteomics data that we may get in KPMP right now: bulk proteomics, subsegmental proteomics, and what has been called near single cell proteomics. So those are those 3 blue columns. And so on the first column that you can see, and I apologize, you probably have to squint to see it, but is those kind of that data pyramid that I was talking about. There's some tissue processing data that we collect, some protein extraction data that we collect, and etc. So then you can see that for each of these different data types, we've decided whether or not it's needed. So for instance, bulk proteomics doesn't need to be presented with the tissue processing properties. So if somebody tries to go into our data lake and upload bulk proteomics data, they would not see those questions. But if they tried to upload subsegmental proteomics data, we would ask them, what was the type of tissue sectioned, when, what instrument, what was the software version that you were using, that you were running. And then you can see there's additional things to say whether or not it's something that's a free text or a drop down. That was a difficult line for us to find of when is it appropriate to have a strict drop down, and when do we need to allow people to expound in a text field, right? Because as everyone that deals with metadata knows, text field is difficult to then later get value out of, but it's also important to allow people that freedom. So here's just on top of this now a screenshot of what that data lake uploader looks like, to just show you that after a user has chosen, say, subsegmental proteomics, this is a little bit of what the form looks like. So we're still, I said, this was first, we're actively really working on this now. Some of the things, we have still some sticky issues to work out with some of the other metadata types. And so we'll continue rolling those out. The beauty of the way that we built this tool, it's very simple for us to, as metadata standards get adjusted, for the tool to be adjusted really quickly along with it. And of course, we certainly also plan on publishing all of these standards for other people, just like we were able to benefit from looking at the links standards. We're hoping that many others can benefit from looking at ours. So that's all I wanted to say. I think we can pause now and take some questions.
Thank you, Becky. That was a very nice overview, and Matthias to tell us about a different kind of metadata and how you're handling it. It's open for questions. We have a few minutes for questions.
Becky, I tried to get to kpmp.org resources, but can't get there. Can you put the link into the chat window?
Sure can, yeah.
I think one key feature I want the group to understand is where KPMP probably significantly different from the HubMap project is that based on the fact that we are focusing on patients with defined kidney diseases, our procurement of the material is largely constrained to renal biopsy cores. They are obtained in a highly controlled manner, obviously after prospective recruitment and consenting of individuals who are fulfilling the enrollment criteria, but that clearly will limit many of the studies we can perform on the tissue, giving the small amounts of tissues. These biopsy cores will be between 14 and 20 millimeter in length and 1.2 millimeters in diameter. So just to give you a starting point, what the available information derived from these tissues will be. And I was very interesting to hear how a common coordinate framework you're currently conceptualizing could actually utilize specific tissue features, particularly on the mesoscale, where these biopsy cores could be reoriented into a larger framework of spatial orientations established for an organ. We will have another challenge that these disease processes probably will alter the landmarks which we can use in healthy kidney tissue to orient tissue inner segments into the existing structural land frames. By altering these landmarks, it might become challenging to reorientate the tissue depending on the level of distortions the disease process has imposed on the tissue.
I had actually a question about that. How much of visual information can you get from needle biopsy? How much the tissue stays together?
Mm-hmm. So we will actually process one entire biopsy core solely for histology using the standard processing steps established for diagnostic renal biopsies, because one task obviously is to detect any relevant information for the disease process to feed back to our study participants. So we will have the panel of standard histological stains available from one entire biopsy core. We will have a second core in OCT where cryosections will be done, and several teams, including the team in IU, will generate then complex images of structural and molecular features of their tissue. And then a third tissue core will alternate in processing, being snap frozen for spatial metabolomics or being utilized for single cell profiling studies using disaggregated tissue from the third core. After disaggregation, we will not have structural information available.
I think this will be very important because other tissues that we might have trouble collecting in toto, we might have to use similar strategies to get some information, but I didn't see any of this, or did I miss it? Did you present, or you didn't have any? examples yet.
Sorry, I don't think I got the context of your questions. Can you please rephrase?
What I was saying first that I think it's important for us to learn what works and doesn't work, because we may have to use similar technologies to collect other tissues that we don't have access to the whole organ.
Yes.
And then I said I would be very interested in seeing how the various technologies work, but I don't think I saw any examples. Did you have?
Yeah, as the procurement protocols, detailed manual of procedures are actually posted on the website as of 6 weeks ago. Please understand that these are processes under intense development, but this is what we have been developed out of unaffected parts of kidney from tumor nephrectomies as a network over the last 2 years. They have shown to be successful on these surgical specimens to generate the various data types. And that information can be downloaded from the KPMP website, and we certainly can make that available via the chat window. Output from these data sets is they are just currently starting to go through the analysis pipeline. And at our in-person meeting last week, we made significant strides forward and are planning to make the first analysis derived from these data available in very short order. And the expectation of KPMP after we finish the ramp-up phase is actually that data generated from the tissue procurement side is posted live after it completed the internal QC steps via various data sharing tools, including the tissue atlas, which Becky has introduced the concept, but also the opportunity to access the raw data files generated from the patients, obviously being aware that we will have some constraints imposed by HIPAA on us on sharing some of the clinical information on an individual level.
Thanks. I think we probably should move on. We are about a minute over the time. Thank you, Matthias. And Becky, please put your questions on the Google Doc sheet that Kati has sent a link. All these questions are very important for HubMap as well as our consortia to deal with. So I think we will next move to the ontologies.